Hello, my name is Hannah Zlotnick, and I will be presenting Fate of Mechano-sensitive Microcapsules after intra-articular injection in a large animal. Osteoarthritis, or OA, is a joint-wide disease associated with pain, inflammation, and tissue degeneration. Intra-articular injections, such as those shown on the right, are appealing to treat the symptoms of OA and/or the disease itself because these injections disperse throughout the joint and limit toxicity associated with systemic delivery. However, free drugs are quickly cleared from hours to days into the lymphatic vessels and venules in the synovium, limiting the efficacy and requiring repeat injections. In an effort to extend drug residence time in the joint space, researchers have investigated various nanoparticle, microsphere, and microcapsule technologies. Microcapsules are particularly appealing because they can deliver a significant payload that is protected by an outer shell. Their size is also highly tunable, and controlled release can be mediated by either degradation of the outer shell over time in vivo or an external stimulus such as mechanical forces, which makes them even more appealing for musculoskeletal applications. Our lab, in collaboration with the Dodge and Lee labs at the University of Pennsylvania, formed these microcapsules using a glass capillary device. The inner aqueous solution contains BSA, the fluorophore Alexa 488, and a drug of choice. The middle fluid, which later forms the outer shell, contains polylactic coglycolic acid, PLGA, Nile red as the fluorophore, and chloroform the solvent. And the outer fluid includes polyvinyl alcohol, or PVA. Here you can see a video of these microcapsules being formed in real time, and over a few hours we can form millions of microcapsules, making this fabrication setup conducive to scale-up. After collecting the microcapsules, the chloroform in the PLGA evaporates, leaving behind the protective shell. Drug release from these microcapsules is mediated by either degradation of the outer shell or mechanoactivation via a mechanical force leading to rupture of the capsules. In our group, we have coined these microcapsules mechanoactivated microcapsules, or MAMCs. We recently published on this technology showing the degradation profiles of different sized MAMCs, where the smaller MAMCs with thicker shells retain their contents for longer. We have also characterized the MAMC mechanoactivation profile, where indirect compression, smaller diameter MAMCs with thicker shells can withstand higher loads. Moving forward, we are interested in clinically translating our MAMC technology for intra-articular drug delivery. Considering the scale from bench to bedside, the typical next step would be to carry out a rodent study. However, since rodent cartilage and the overall size of mouse and rat knees is much smaller than the human, we selected a large animal model in a Yucatan mini pig, which better mimics the loading of the native human joint. The purpose of our study was to assess retention, localization, and integrity of MAMCs after intra-articular delivery in a large animal. And we hypothesized that these MAMCs would withstand injection, be retained in the joint space, and progressively rupture over time in vivo. For this study, we fabricated fluorescently labeled MAMCs and injected 500,000 MAMCs per knee using a 21-gauge 1.5-inch needle. We had two cohorts of animals, where cohort 1 received an injection 14 days before euthanasia, and cohort 2 received an injection 1 day before euthanasia. Here you can see our anterior lateral injection that we performed on each animal. Post-euthanasia, we performed IVUS imaging to look at joint-wide microcapsule retention and fluorescent microscopy to look at the integrity of the MAMCs. The MAMCs used in this study had an outer diameter of 34 microns, which is similar to the smallest batch of MAMCs from our previous studies. Before moving on to our in vivo work, we wanted to ensure that extrusion through a needle would not cause rupture of the MAMCs. Here you can see that there was no difference between MAMCs injected through a 27-gauge needle and MAMCs pipetted onto a glass slide. At the terminal time point for each animal, we utilize the left injected knee and the right no injection control for IVUS imaging. Here you can see a sample limb within the IVUS spectrum machine. We imaged each of these knees in 4 planes, including the anterior, posterior, lateral, and medial aspects. To analyze these images, we calculated both the total signal area and peak radiant efficiency. Within Cohort 1, which received an injection 14 days before euthanasia, we are excited to see that the MAMCs were still detectable by IVUS at this later stage. And as expected, within Cohort 2, which received an injection 1 day before euthanasia, we see strong signal via IVUS. Quantification of these images shows that the radiant efficiency and area of signal decreased over MAMC time in vivo. After IVUS imaging, we carefully dissected the joints and imaged individual tissues, showing accumulation of MAMCs throughout the entire joint. Here you can see the adherence of MAMCs to the patellar tendon, adipose tissue, meniscus, and trochlear groove. We then zoomed in to look at the percentage of intact microcapsules over time in vivo. We first established a baseline by injecting MAMCs into cadaveric Yucatan stifle joints, and we found that around 93 to 95% of MAMCs were intact at time 0. As expected, there was a decrease in the percentage of intact microcapsules from time 0 to day 1, and again from day 1 to day 14 in vivo. Taken together, we show that MAMCs are a promising drug delivery platform for the knee joint. We can safely inject these capsules, which are then retained within the knee and progressively release their contents over weeks in vivo. While for this study we only utilized one size of MAMCs, this technology is highly tunable, and future studies will pursue the administration of heterogeneous populations of MAMCs for sequential drug release. Future studies will also investigate the delivery of MAMCs to a degenerative joint. For this work, we will utilize our lab's large animal model of osteoarthritis, whereby we destabilize the medial meniscus. And we will fabricate MAMCs with bioactive agents to prevent degeneration in the presence of cytokines. We have preliminary evidence to suggest that MAMCs adhere better to damage versus healthy cartilage, and we look forward to establishing the efficacy of this drug delivery platform for osteoarthritis. With that, I would like to acknowledge the MAMC team, Dr. Ching-Hui Huang, the IVUS Core Director, the ULAR staff, the Mock Lab and Diamond Lab members, and my funding sources. Thank you.